Hi everyone, myself Bilas. I welcome you all in my YouTube channel Bilas Zoology. Today we will discuss the topic histology of kidney. But before coming to the main topic, I would like to discuss the location and physiology of kidney. Excretory system of vertebrates including mammals is formed of one pair of kidneys, one pair of ureters, a urinary bladder and a urethra. Actually, kidney are primary or major excretory organs of man and other vertebrates. Those are one pair, large size, approx 10 to 12 cm long, 5 to 7 cm broad and 2 to 3 cm thick. It's like bean-shaped, dark red, slightly flattened structures present in upper part of abdominal cavity between the levels of last thoracic and third lumbar vertebra along dorsal body wall. One on either side of vertebral column. Concavity of kidney called hilum or hilus renalis is always inward directed. These are asymmetrical in position. Right kidney is slightly posterior in position and is probably pushed by well-developed liver. These are extracoelomic and metanephros in position. Internally, each kidney is formed of about 1 million uriniferous or renal tubules or nephrons held together by connective tissue and surrounded by a tough white fibrous capsule which is further surrounded by a shock absorbing layer of fat called renal fat, followed by a fibrous membrane called renal fascia. Nephrons are convoluted in outer part of kidney called renal cortex, while run straight in a central part of kidney called renal medulla. The renal medulla is with 8 to 18 conical masses called medullary pyramids, which project into minor and major calyx. The renal cortex extended in between the medullary pyramids as renal columns called columns of Martin. These respectively give dotted and straighted appearance. Now, I would like to discuss the Bowman capsule. Actually, it is a double walled cup present in the renal cortex. It is lined by a thin and semi-permeable squamous epithelium formed by flat cells. Inner and outer layers of Bowman's capsule are respectively called visceral layer or parietal layer. The visceral layer of Bowman's capsule is formed of special epithelial cell called podocyte or foot cell, having pit-like structure, thousands to millions of minute pores called silt pores for the passing of ultrafiltrate. In the cavity of Bowman's capsule, there is a group of about 50 capillaries called glomerulus Glomerular capillary endothelium has numerous minute pores called fenestrae. Between the visceral epithelium and glomerular capillary endothelium, there is a basement membrane which acts as a dialyzing membrane. The visceral layer, basement membrane, and glomerular epithelium collectively called filtering membrane. It acts as an ultrafilter and is responsible for ultrafiltration. So, permeability of glomerular membrane is formed 100 to 500 times as high as that of the usual capillary. Actually, Bowman's capsule and glomerulus combinedly they call Malpighian body or renal corpuscles. Now, if we have closer look, we will observe that actually kidney enclosed in a capsule of connective tissue. Kidney consists of an outer granular cortex and a radially striated inner medulla. Medulla consists of a number of conical masses, the renal pyramids. The base of the pyramid are adjacent to the cortex and apices. The renal papillae point towards the renal pelvis. The pelvis is extended and forms the beginning of the ureter. Closely packed uriniferous tubules are present both in cortex and medulla. The amount of interstitial connective tissue is small in the cortex and more in the medulla. The functional unit of kidney is the nephron. A nephron consists of renal capsule with glomerulus proximal convoluted tube, loop of Henle, which is divided into descending and ascending planes. Leaving medulla, the tubule enters cortex as distal convoluted tube near renal capsule. The tubule is connected with a collecting tubules through a short junctional tubules. The collecting tubules receives other junctional tubules and several of the form of papillary duct. Now, to identify the cross section of the kidney, we can observe the staining area. We will identify that it is made up of two layers, an outer deeper stained cortex and inner lighter stained medulla. Cortex showed many scattered dark stained round renal capsules. Connective convoluted tubules of different diameters and pale staining radiating lines, that is the medullary rays. Each renal capsules consist of large irregular shaped masses, that is the glomerulus, a distinct Bowman capsule space and thin walled Bowman's capsule. Convoluted tubules are of two types, proximal and distal. Proximal convoluted tubules with large deep stained cells with small lumen. Distal convoluted tubules with small pale stained cells with white lumen. Medulla is a dense mass made up of three types of tubules. Henle's loop with small diameter, collecting tubules having large diameter with deeply stained cells, collecting duct of large diameter with lightly stained cells. In this way, we can able to identify the cross section of kidney. Thank you everybody. Please like, share, comments and subscribe my channel and press the bell icon. I am Bilas and you are watching my YouTube channel Bilas _Zoology. Thank you everybody.